Hello everyone, welcome back to another video from Shomu's Biology. And in this series of lectures, we are talking about the DNA sequencing methods. So today's discussion is another type of DNA sequencing, and today we're going to talk about 454 sequencing, 454 pyrosequencing. Pyrosequencing, okay. Uh, 454 sequencing, pyrosequencing is also called as Roche sequencing, whatever term you use. These are all the same method, same technique of sequencing a DNA. Now, to give you a basic idea about it, 454 sequencing is advanced mode of sequencing than the Sanger sequencing that we had. The Sanger sequencing is the easier mode and only to be applied for the DNA sequence up to 1000 base pairs long, while 454 sequencing can be used for sequencing bigger DNA segments, bigger DNA area. And also 454 pyrosequencing is faster than the Sanger sequencing, but it is not cheaper, it is expensive. And in the recent years, we have advancement of DNA sequencing technology, and we use Illumina sequencing, which is even faster than 454 pyrosequencing and comparatively cheaper than 454 pyrosequencing. So Illumina sequencing kind of replaced 454 pyrosequencing, but for subjective knowledge, for your exam preparation and your overall semester preparation, you need to know what 454 pyrosequencing was. So here we are to talk about 454 sequencing, which is kind of outdated now, but we need to understand it. Now, in case of 454 pyrosequencing, we are trying to find out the DNA sequence with the help of, let me write it, with the help of light. A guiding light will help us to get it, plus some chemicals. Okay. So we can get a sequence of a DNA by using light and chemicals and beads. Okay, beads. Now, when we talk about 454 pyrosequencing, I always tell the students to think of one thing, is that we our whole genome is built with the DNA is built with this. It's just like a locket. It's just like the 4-bit sequence that are out there: A, G, C and T, right? And these are the letters of the DNA, and we need to know which letters are positioned after what and at what number. That is known as the sequence. That's what we need to understand, right? So in case of 454 sequencing, we'll be using light, we'll be using some chemicals, and we'll be using beads. Now beads are made with multiple things, but example is streptavidin is one example of the component that makes the beads. Now for this particular process of 454 sequencing, what we do here is basically we start this process of breaking the DNA, because our target DNA is really big, right? Because for a smaller target DNA we will not use 454, we will always use the Sanger sequencing. So larger DNA is fragmented and smaller fragments are generated. So DNA cut and fragmentation is the step 1 on this process. And once that fragmentation is done, it's not enough. Then adapter ligation is another very important step. Ligation of adapter. So we'll take one of these DNA fragments and we will see the adaptation of the ligation of adapters. Okay, we'll add 2 adapters. Let's say adapter A and adapter B, with 2 adapters are added to it, right? Among them, one of the adapter is designed in such a way that it can adhere to or stick to the beads that we are going to utilize. Based on that, the adapters are designed. And what are the adapters? What are these things? These are also made with DNTPs. DNTPs. Basically, these are small stretch of nucleotide sequences as adapters. Okay, but these adapters can interact and can adhere to the bead. That's how the adapters are designed. Okay, so the bead that we are going to talk about, the bead is a place. Where this, with the help of the adapter, let's say, let's draw this the blue color adapter here, and this is our target DNA in black. Okay, and the red other adapter is placed here like this. Okay, so this is the bead. So let me write it here. This is the bead. Streptavidin is an example of the bead. Now with this bead, this adapter A, the side of the A adapter is fitting in. So basically our target DNA is fitting into the bead, right? It's sticking to the bead like a glue, right? It's sticking there like a glue. So that's the step number 1. We fragment the target DNA into smaller pieces, then we add adapter, and then it comes the attachment of the beads. You can call it step 1 or you can call it two steps. One is the step 1 is the break and the DNA fragmentation and adapter ligation. Step 2 is where the adapter ligated content is transferred. It is transferred to the beads. So now what we have here, the bead is associated to the adapter associated or adapter ligated target DNA content. Okay. Now the beads are very tiny, and individual beads will carry individual DNA fragment. So basically one bead, one DNA fragment. That is the rule. That is the rule. One bead, one DNA fragment. Okay, and we create that rule. We continue with this rule. Once this beads associated to the target DNA with the adapter, then what we need to do is basically before sequencing, we need to get rid of both the strands of the DNA, because here we are looking at double stranded DNA, right? These are the basic information that nobody is going to tell you. Now here we need to make it single stranded, right? So separation of the strands are done, and that is done by alkaline treatment. Alkaline treatment helps us to separate the strands from each other, and ultimately what we end up in these beads. Let me draw again. We have the beads and we have our DNA is attached to it with the help of the adapter. Let's say the blue adapter facing this side, and rest of the black, rest of the red. And let me draw it like this so that you will understand that this is a single stranded DNA that is attached to the beads. So that is the ultimate orientation that we get. Now once we get that, we need to make more and more copies of the DNA, right? So we need to make it more copies. So for that, what we do, we use oil. We use oil emulsification here. So basically we cover the bead associated with the target DNA with oil. So oil coating is done, and a droplet is formed. A oil droplet is formed, and inside the droplet we also add some components. We also add components for the process of polymerization or making copies of these DNA that is added to the bead. And who will copy the DNA? Via the method of polymerase chain reaction or PCR. And as we are using oil droplet and whole machinery of cloning and copying inside the oil droplet, we call it as a emulsion PCR. Emulsion PCR. Okay, emulsion PCR process is done. Basically DNA will be copied using polymerase chain reaction, but in this case we are doing it inside the oil droplet. That's why we call it emulsion PCR. Once the emulsion PCR is done, we have multiple copies of this. So multiple beads with multiple copies of the DNA. So huge number of copies of the same content is created with the help of our emulsion PCR. That's why the PCR is done, to make more and more copies, because when we start working with a fragment of the DNA, we want multiple copies of that same fragment, and to achieve that we use the emulsion PCR for that. Once that is done, we will use wells of plate. So plate with wells in it, and multiple wells are present in there, and these beads containing the target DNA will be loaded into the wells. Okay, so it will be loaded into the wells. So basically we have this glass slide. Let's say this is the place where multiple wells are present. Okay, multiple wells are present, and in those multiple wells we will load. So loading is done. Loading will be done. So loading of what? I'll show you here. Basically the loading of the beads associated to the target DNA is loaded into each of the well, and once we load it, so load the bead containing DNA is loaded. Along with that, we also put the DNA polymerase The target DNA here is double stranded, single stranded obviously. The DNA polymerase is added and DNTPs are added, okay, and primer will also be added. Primer will also be added, right, inside individual wells. Inside individual wells, in individual wells, we will add DNA polymerase, DNTPs and primers. We will add all these necessary components, all these necessary chemicals and factors and proteins for the process of synthesizing new strand. And again here we will apply the same logic that we applied in most of the DNA sequencing techniques. We are simply try to do a DNA replication. We try to make a copy of the DNA using the target DNA as a template. The target DNA, the template DNA is the DNA that we are about to sequence, right? So we are trying to sequence it by adding new nucleotides complementary to the nucleotides that are shown by the target DNA in there, right? That's what we are doing in this case. So we will load everything here, okay? In this particular case, now what will happen is something. This is where inside the well the real magic will happen. And inside the well what we have, let me draw it from the beginning here. We have the bead, remember, and we have the receptor for the beads, and we have this single stranded addition. We have the sequence of the DNA, right, and rest of the adapter. And with the help of this adapter, let's say we add the primer, okay, and this is primer, let's say, and we have an extension of the primer, right? Now we are trying to extend, right, this 3' hydroxyl area we are trying to extend. While extending it, there is this sequence, there is this black colored sequence, and let's look at this sequence something like this. Let's say A, let's say T A C C A G G T A. Okay, so this is the sequence. Let's say we are zooming in this sequence here. If this is the sequence and we have DNTPs added, now the DNTPs that we add, they are not modified, they are not they don't contain any fluorescent dye, they don't have any DDNTPs, nothing like that. It's normal DNTP, right, and everything that we provide in here. But what we are doing here, we are adding one type of nucleotide at a time. We will either give A at a time, then we wash the wells, then we add G, wash the wells, then we add C, wash the wells, then we add T, wash the wells. So we'll continue to do this, but at a particular time we will provide the well with only one DNTP, whether ATP or GTP or CTP or TTP, any one particular type. Let's say we start with ATP, right? Let's start with ATP. I'll take a different color. ATP. We'll start with ATP. Now ATP, A binds here, so that's the addition. Good, we'll get a response. Now how do you get a response? Now we need to understand. Once A is bound, because this is the correct pairing between A and T, so we get a signal. How do you get a signal from a normal DNTP which is not fluorescently tagged? We use light. Remember I told you, we use light coming out from the beads to detect the presence of a particular nucleotide. How do you do that? Now when a successful interaction is done, right, when a successful interaction is done, in this well we also add what? Whenever a successful addition of nucleotide is done, it releases PPI, pyrophosphate. Pyrophosphate is released by a normal chemical reaction. So ATP, when we provide them, it gets attached, right, AMP in that form, and two other phosphate will be released as PPI. That's a normal process of DNA polymerization. Now this pyrophosphate, PPI means pyrophosphate. Now in this well, after the addition of nucleotide, what else we provide? We provide APS. Okay. APS is provided as a reagent. APS is a chemical reagent. What APS does is that APS converts PPI into ATP. And we also provide luciferin. ATP once in contact with luciferin is going to generate light in presence of luciferase enzyme. So remember, inside the well, how many things that we provided. Remember I told you 454 pyrosequencing utilizes light and chemicals to give us the sequence of the DNA. Here comes the light. Light gives us as a response to tell that yes, correct binding of nucleotide is done. But as a chemical, we need to give APS, we need to give luciferin, we need to give luciferase. All this we need to provide, right? So ATP will be converting into light, right? And that is possible when luciferase is present, converting luciferin into light, and APS is required to convert pyrophosphate into ATP, right? So this is the sequence of reaction that you always need to know, a sequence of reaction, and this is where the name pyrosequencing comes in. This is why pyrosequencing comes in, because this luciferin associated to ATP becomes oxyluciferin and visible light is emitted, and whenever there is visible light we can always find it out easily. It's easy to find out. We don't need expensive machines to find out light, right? Existing machines can do that. Now in this case, this APS, adenosine 5' phosphosulfate, it is known as adenosine 5' phosphosulfate, APS, adenosine 5' phosphosulfate, and they use the enzyme ATP sulfurylase that converts the ATP sulfurylase, that converts the PPI into ATP. That's the energy molecule. In this case, it's not likely the ATP is generated because A was there. If it was G, if it was C correctly pairing, the same thing happens. Now let's get back to the situation and try to reconnect the dots. Now once that is done, we'll wash it off, then we provide the next. Let's say we give the next one as C. The C will pair with A? No. As the C will not pair with A, so there won't be any reaction, there won't be any light. Then let's say we give G. G pairs with A? No. So won't be any light. Then we give T. T pairs with A? Yes, T pairs with A. And again when T pairs with A, a correct pairing that again triggers the release of PPI. That PPI will be converted to ATP by ATP sulfurylase converting to ATP. ATP associated to luciferin will be converted to oxyluciferin by luciferase enzyme that will generate light. So again if T pairs with A, which is a correct pairing here, that gives us a light. Next again we repeat the same thing. We start with A connecting, next one is C, so A will not pair with C, no light. Then we go with G. G pairs with C, again light, right? Again wash, start with again A pairs with C, no light. Then again G pairs with C, light. Okay, then again starting with A pairs with A, no light. Then again G pairs with A, no light. Then again C pairs with A, no light. T pairs with A, yes, light. So by every single rounds we are running this whole process. For every single round of any single base, we are running it for A TP, we are running it for GTP, we are running it for CTP, and we are running it for TTP. For all the normal bases we are running it, and if there is any base pairing, then this base pairing, remember if there is base pairing, it's going to generate light by the chemical reaction that we have talked about. In the chemical reaction we gave APS, adenosine 5' phosphosulfate, we give the enzyme ATP sulfurylase, we give luciferin, we have the production of oxyluciferin, ultimately production of light in presence of luciferase. So all these chemicals along with the normal DNTPs need to be provided, along with DNA polymers need to be provided inside the well. So in individual tiny wells, all this complicated chemical machinery is happening. It will continue, and whenever there is a proper base pairing, it will signal it by generation of light, which can be detected by the detector. And whenever we detect the light, that means that particular base. So we are running for A base. If there is light, that means what? A pairs, right? And when there is no light, A does not pair with that complementary sequence. This process continues, right? And it not only gives us the light but also the light intensity. If there are back to back 2C were there here in this case, right? So the light intensity, remember, the light intensity will be high. The light intensity will be little higher in this case. If there are consecutive 4 same nucleotides, again the intensity will be little high. But for light, intensity detection is not that easy. So sometimes when there are, let's say, 20 stretch and there are 18 stretch, it is not easy to differentiate between the 15 stretch of the same nucleotide sequence to the 20 stretch of the same nucleotide sequence. So that will be a tight call. So those are some ambiguities that can be found in the 454 sequencing. For that, a drawback, we no longer use it. Besides, we have a better cost effective model. We have Illumina sequencing, which is comparatively cost effective, comparatively less pricey than 454 sequencing, and also less chemical involvement is in there. So considering these two facts, the DNA sequencing methods that we use nowadays are primarily Illumina mode of the sequencing that we use. But 454 sequencing is good. It's better over the Sanger sequencing because it can sequence larger segment of the DNA, and it is much faster than Sanger sequencing. Compared to Illumina, this one is slower than Illumina. Illumina is faster, Illumina is cheaper, but 454 has longer or lengthier reading frame. That's why 454 still can be used in some cases if your reads are lengthier. In case of 454, the reads, remember the reads here near about 500 to 700 base pair reads can be easily sorted or easily sequenced. In case of Illumina sequencing, the reads are 300 to 400 base pairs, but in this case 700, 800 base pair read can be easily sequenced with good high accuracy rate with the help of 454 pyrosequencing. Why it is called pyrosequencing? Because it uses this pyrophosphate that is generated as a result of the new nucleotide addition and conversion of that ultimately to visible light. So basically lights shining on top of the beads. So whenever we see light shining on top of the beads, we already get it. So if the light's shining, the proper binding happens, right? Sequence is known. If no light, no binding, so that is blank. So either the sequence or the blank. With this idea, then our computer can analyze the sequence and give us the full data about the sequence. That is all about 454 pyrosequencing. I believe you have a clear idea on 454 pyrosequencing now. If you get this idea clear, then also watch the other DNA sequencing lectures of this series, and like this video, subscribe to this channel, share this video with your friends so that they also get to know about this. Okay, thank you everyone. I'll see you in the next video. Bye.